Hello and welcome back to Biology for Beginners. Last week we finished up Chapter 9. This week we're going to be diving into Chapter 10, the molecular biology of the gene. Now this chapter is a little bit heavy with material, so our sections are going to be a little bit shorter here. But that doesn't necessarily mean the videos will be shorter, and we will see why. This week we're going to kick it all off by talking about the structure of genetic material. As always, before we jump into the material, right now you should see those terms and definition slides popping up around here-ish, so you can grab the vocabulary we will be using today. Also, a quick reminder, if you want the entirety of my PDF slideshow, go ahead and pop on over to my Patreon. The link is in the description below, so you can grab that for absolutely free, but anyone who does sign up to be a patron will get these videos, all study materials, and some other things early as well. I'm still working on that a little bit because life is messy since I'm finishing up college in a few months. But without further ado, let's get into this. Today we need to begin with a little bit of history before we get into the nitty-gritty of the molecular biology here. So back in the early 20th century, what biologists knew were that genes were located on chromosomes. And chromosomes were made up of DNA and protein. However, what biologists did not know was if DNA or the proteins on the chromosomes were what was actually carrying the genetic material. So what exactly did they suspect? Well, they suspected that proteins would probably be determined to be the genetic material when they began experimenting because proteins are more complex than DNA. And their reasoning was that there were 20 known amino acids that made up the proteins versus the 4 known nucleic acids that made up the DNA. So, logically thinking, genes are fairly complex, so the more complex molecule would probably be the ones that served as the genetic material. So, that makes sense logically and intuitively, but as we know here, science doesn't rely on intuition. So, moving to 1928, Frederick Griffith attempted to create a pneumonia vaccine, and he was doing this by working with 2 strains of bacteria, one that was pathogenic, so one that actually caused the disease to pop up, and one that was harmless. And he found that when he mixed the dead pathogenic bacteria with the living harmless bacteria, some of the living bacteria became pathogenic, and their descendants could also cause the disease. So that is actually the experiment that started the race to figure out what genetic material actually is. Is it DNA or is it the protein? Now, fast forward to 1952, and we get to where Alfred Hershey and Martha Chase were experimenting on T2, a bacteriophage that specifically infects E. coli. A bacteriophage is just a virus that infects bacteria. You'll also hear it referred to as just phage. And what their experiments did was that it showed DNA was the genetic material of T2. Prior to the experiment, they knew that T2 could take over and program the host to make more T2. That's how viruses reproduce, and that's also one of the reasons why viruses aren't technically considered living, because they can't reproduce on their own. They have to use another organism to make more of that virus. If we look at the image here, we will see a TEM of T2 and also a diagram of T2. The important thing to notice is that that outside yellow part is primarily just protein, and then the little cutaway where it's showing the blue squiggles is the viral DNA. But before running this experiment, what they didn't know was if it was DNA or protein that was used to take over the host and cause it to make more T2. Before we move into the actual experiment, let's talk a little bit about that virus life cycle, specifically the bacteriophage life cycle. So in step 1, that phage is going to attach itself to that bacterial cell. In phase 2, the phage is going to inject that DNA. Remember, before the experiment, they didn't know if it was protein or DNA. And Phase 3, the phage DNA is going to basically hijack what is in that bacterial cell, making the cell do the cell things, and cause it to make new phage parts. So the proteins and the DNAs, and then those parts are going to get put together. Once a bunch are made, the cell is going to lyse or explode basically, and the new phages will be released. The cycle will start again. So what exactly was the experiment? Well, it was a fairly simple experiment, and they had 2 batches that they did. So in batch 1, they took a radioactive protein and kind of dyed it. So what it does when you use these radioactive things in biology experiments is it helps you to see the thing that you're looking for, because it's very easy to detect the radiation. So they took the radioactive protein, which is in yellow in this diagram, and they basically made all that phage protein radioactive. They allowed it to infect a bunch of bacterium and then kind of mixed everything up, ran it through a centrifuge, which basically just spins things very fast and separates layers, which caused the bacterial cells to kind of form these little pellets. And then they measured where the radioactivity was. Was it in the pellets or was it in the liquid? And in the case of the protein, the radioactivity was in the outside liquid. The radioactivity was not in the pellets at all. So move into batch 2, where the radioactive portion was actually the DNA, and the actual experiment was pretty much exactly the same. Allow those phages to infect the bacteriums, centrifuge everything, and then measure where the radioactivity was in the bacterial pellets or in the liquid where the proteins were. And in the case of the DNA, the radioactivity was in the pellets. So when they put everything from this experiment together, since the radioactivity was only in the pellets when it marked the DNA, that basically meant that DNA was the genetic material and the protein was basically just there to carry it and host the DNA. So what exactly is DNA and RNA while we're at it? Well, if we think all the way back to the molecules of life, you might remember we talked a little bit about nucleic acids. And a nucleic acid is a polymer made up of covalently bonded monomers. Monomers are just a smaller unit that gets bonded together to form a polymer. And in this case, the monomer is called a nucleotide. A nucleotide is made up of a 5-carbon sugar, so deoxyribose in the case of DNA, or ribose in the case of RNA. A nitrogenous base, which we will talk about all those bases in just a few moments, and a phosphate group. So the nucleotides are a fairly simple thing. They get stuck together to form a polymer that makes the nucleic acid. DNA and RNA are both nucleic acids. And how those nucleotides are actually formed together to make that nucleic acid is that the phosphate group of one nucleotide will bond to the sugar of another, and that will end up forming what is known as the sugar-phosphate backbone. You can see that on this image in the part that's kind of colored blue and yellow. But if we move on and kind of stretch that coil out, it's a little bit easier to see. And on this slide, what we're looking at are different representations of DNA as the polynucleotide or the polymer that it is. So if we notice that blue and yellow represents that sugar-phosphate backbone, with the blue representing the sugars and the yellows representing those phosphate groups. The sugars are much larger than the phosphate groups, which is why they are kind of not quite the same size there. And then you can see the different nitrogenous bases kind of sticking out from those sugars, and they're labeled here as A, C, T, and G, which we will get into what that means in just a moment. So If we'll notice here, just kind of zooming in a little more on that little square, you can see a single nucleotide here, that sugar, the phosphate group, and the nitrogenous base are all kind of bonded together covalently. In this case, the nitrogenous base is thymine. But for the sake of being thorough, I've also included an RNA nucleotide as well, because they do have slightly different sugars. And so in the DNA nucleotide, you'll notice it's labeled deoxyribose, and in the RNA nucleotide, it is labeled ribose. We'll get a little bit more into that difference in just a few minutes. And the nitrogenous base here for the RNA nucleotide is uracil. So let's talk a little bit more about the nucleotides. And first, we're going to start with the primidines. Now, primidines are characterized by that single ring that you see. If you notice, the carbons and the nitrogen in this diagram form one ring, and that is very important. That's pretty much what makes it a primidine. So the primidines are going to be thymine, which is actually not present in RNA, it's only present in DNA, and in RNA, thymine is replaced by uracil. And thymine and uracil both attach to adenine. And then the other primidine here is cytosine, and cytosine attaches to guanine. So now that we've talked about the primidines, let's move on to the purines. And while you notice that the primidines had one ring, the purines are characterized by a double ring, which is easy to see if you're looking at them. You can see that the carbons and nitrogens form two rings. And our purines here are adenine, which can attach to thymine or uracil, depending on if we're looking at RNA or DNA. And then guanine, which attaches exclusively to cytosine. So what's the difference between RNA and DNA? Well, DNA, the sugar is going to be deoxyribose, which is why DNA is short for deoxyribose nucleic acid, and RNA, the sugar is ribose. So RNA is short for ribose nucleic acid. And then if we're looking at the nitrogenous bases, DNA has thymine as a possible nitrogenous base, and RNA has uracil instead of thymine. Those are the big differences here. There are more differences when we talk about the specific functions, but we're going to get into that in a later video. So just to kind of highlight the difference between deoxyribose and ribose, the two sugars are shown here on this slide, and what I have circled is the difference, and it's a very subtle difference, but it's the thing that literally defines the sugar. So deoxyribose, you'll notice, has that carbon bonded to two hydrogens, whereas at that same carbon in ribose, it's bonded to a hydrogen and an OH group. So what the deoxy and deoxyribose means is there's no oxygen there. Deoxy, no oxygen. That's the difference, is an oxygen. Deoxyribose is lacking the oxygen and ribose still has it. You get a little more into what it does to the structure and stuff if you take organic chemistry, but we're not going to get into that here. The important thing to know, if your bio professor is even asking you to know it, is that deoxyribose lacks an oxygen and ribose has it. So if they show you the picture and ask which one's deoxy, which one's ribose, you'll be able to tell them. But most entry-level bio classes that I know of, that I've taken, don't really get into the difference. They just want you to know DNA is deoxyribose, RNA is ribose, but they don't really get into the difference. Now let's talk about the DNA double helix, because that is the thing that even if you're not a big bio person, you can usually recognize as DNA, even if you don't know what the nitrogenous bases are, even if you don't know what DNA stands for. So in 1952, like we talked about, the Hershey-Chase experiment determined that DNA was genetic material, not proteins. And after that, the race to determine how that DNA structure actually accounts for the role in heredity began. And around this same time, I don't have the exact year, Rosalind Franklin produced an X-ray image of DNA, and that image was seen by a biologist, James D. Watson. Now we can see the picture here is of Rosalind Franklin and her X-ray image of DNA that kind of sparked all this that we're going to talk about in just right now. So James D. Watson and Francis Crick are responsible for the model that we know of that helps us understand the structure of DNA. And so what they did was they constructed quite literally a wire model of DNA based on the known chemistry of DNA. So they knew what the nitrogenous bases are, they knew what made up DNA. And Franklin's data, because she had been working on DNA and other molecular things for a little bit at that time. So based on Franklin's data, they knew that the sugar-phosphate backbones must be on the outside and the nitrogenous bases must be on the inside. And so they also, after like trial and error and a lot of different things, the DNA was uniform size based on the imaging that they saw, which means that while they originally thought that primidines and purines paired with like, so a primidine would pair with a primidine, a purine would pair with a purine, that wouldn't be possible because of the single and double rings. It would not have the uniform size that they knew it had. So that meant that primidines must pair with purines. They did not pair like with like. Now, after a lot of trial and error, they figured out the ATCG pairing. So that is important because they figured out that that is exclusive, that A will never pair with C, G will never pair with T. It will always be ATCG. And so if we just kind of look at this diagram here, we can see that. And these are some common things that you'll see when you're learning about this stuff. On the left, we have the classic 2D ribbon model, which you'll see, which shows that pairing. And the pairings we see here are just the classic shapes that they use to illustrate how these things pair together. Because C and G complement each other, they have that more rounded shape to them. And because A and T complement each other, they have that more pointy shape, which just kind of helps drive home that these things go together like little puzzle pieces. They can't be paired in a different combination. And then we have the partial chemical structure, which is a more 3D-ish model that kind of shows you where all the bonds are and things. Because the bonds between these bases are actually hydrogen bonds. They're not covalent bonds. So they're a very weak bond that can be pulled apart. And that's important for DNA replication and reading the information on the DNA that we will get into in later videos. And then if you want a more complicated model, we have the computer model here, which usually you're going to see in biology textbooks because it's kind of cool to look at. So that's all we really have for today. That's a lot of information for one video. But the big takeaway here is just kind of knowing the structure of DNA, knowing that it has that sugar-phosphate backbone, knowing what a nucleotide is, and knowing those nitrogenous bases, knowing that A pairs with T and C pairs with G. Now, in the case of RNA, A will pair with U, but we'll get into that in a later video. And while the pairings of the nitrogenous bases are exclusive, and you can look at one strand of DNA and know exactly what the other strand looks like, they can be ordered in any possible way, which is why 4 nitrogenous bases can code for so many different genes and be responsible for so much genetic variation. So now that I promised I would end this video and then threw more information at you, I will end it for real now, and I will see you all in the next one.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.